Okay, welcome everybody. This is going to be the back end of the chapter on digestion, chapter 24, and what we're leading off here with is the anatomy of the liver. Now, the liver is a pretty vital organ. It has many roles in the body, detoxifying the blood and partitioning the nutrient supply as it comes from the gastrointestinal system are two of the major roles of the liver. It also secretes bile, which helps to emulsify fat in the duodenum so that it can aid in lipid digestion. It's wrapped in a tough fibrous capsule, as are many of the internal organs, and it's covered by a layer of visceral peritoneum. Now remember, when we're talking about peritoneum, we're talking about a membrane. Here, when we say membrane, we mean a sheet of tissue. And when we're talking about the visceral peritoneum, we're talking about the sheet of tissue that lies closest to the organ. On the anterior surface, the falciform ligament marks the division between the organ's left and the right lobe. A thickening in the posterior margin of the falciform ligament is the round ligament, or ligamentum teres, and this is a fibrous band that marks the path of the fetal umbilical cord. So essentially what this is a leftover of is the ductus venosus, okay, this falciform ligament right here. All right. What happens at birth is that due to pressure changes in the circulatory system, this what used to be a blood vessel collapses and becomes a tough ligament. On the posterior surface of the liver, there's an impression left by the inferior vena cava, and this marks the division between the right lobe and the small caudate lobe. Okay, so again, looking on the posterior surface, there is the right lobe, and the caudate lobe is this area right here, okay? Inferior to the caudate lobe lies the quadrate lobe, all right, so locate the quadrate lobe right there. And the quadrate lobe looks like a quadrilateral, remember from your geometry, a four-sided figure, okay? It is sandwiched between the left lobe and the gallbladder. There's the left lobe, there is the gallbladder. Afferent blood vessels and other structures reach the liver by traveling within the connective tissue of the lesser omentum. And this is a common theme that we've seen in the connective tissue throughout our look at the digestive system. It always serves as a passageway for nerves and blood vessels and lymphatics. They converge at a region called the porta hepatis, known as the doorway to the liver. The doorway to the liver. About a third of the blood that the liver gets is arterial blood from the hepatic artery. The rest is venous blood from the hepatic portal vein, which begins in the capillaries of the esophagus, the stomach, the small and large intestine. Hepatocytes adjust circulating levels of nutrients through selective absorption and secretion. Essentially what happens here is that the blood that's drained from the GI makes its first stop inside the liver, and what the liver does is control the chemical composition of the blood before it rejoins the circulation at the inferior vena cava up here. Blood leaving the liver returns to the systemic circuit through the hepatic veins, and this opens into the inferior vena cava. So all the blood from the GI stops off at the liver. The liver then either takes the nutrient material and stores it as fat or as glycogen, or if the body's energy needs are relatively high, it lets that nutrient material rejoin the circulation in general through the inferior vena cava to the rest of the body. And it also detoxifies the blood as it flows through the sinusoids that are in the hepatocytes that we'll see coming up next. So a lot of roles here for the liver. And this is what liver tissue looks like. It's kind of unusual looking. It's arranged in these hexagonal patterns. Each liver lobe is split by connective tissue into about 100,000 lobules, and these are the functional units of the liver. Okay, so what you're looking at there is a lobule. Okay, notice something. Just look at the lobule for a minute, and what do you see? You see these unusual crevices running all through the lobule. You'll also see an arrangement of vessels at each corner of this hexagon, which is called a hepatic triad, and it's made up of a bile ductule, a vein, and an artery, and then there's also a central vein in the middle of this. Okay, so think about that as we talk about the function of these lobules. Each of these lobules is about 1 millimeter wide. Adjacent lobules are split from each other by an interlobular septum. The hepatocytes, which are these sort of cuboidal-looking yellow cells in this figure, they're what do the cleaning of the blood. The hepatocytes in the lobule form a series of irregular plates, and they look a little bit like spokes on a wheel, kind of, right? You can imagine that that's an unusually shaped wheel. In addition to containing endothelial cells, the sinusoids, which are these irregular spaces that run through the lobules, includes a large number of Kupffer cells, and we'll talk about what the Kupffer cells do in a minute. Let's move deeper into the anatomy. Blood enters the liver sinusoids from small branches of the hepatic portal vein and the hepatic artery proper. If you look up in lab at the liver model and you look at the back side of the liver, what you'll see is a purple vessel that enters the liver, and that is a mixture of the blood that's draining from the GI and also from the hepatic artery. So the blood that's draining from the GI is deoxygenated. The blood in the hepatic artery is oxygenated, and when they mix, they form semi-oxygenated blood that's also very rich in nutrients. A typical liver lobule has a hexagonal shape and cross-section. There are 6 portal areas or triads at each corner of the lobule, and you can see them, right? There's one at each little vertex there. The portal area contains 3 structures: a branch of the hepatic portal vein, a branch of the hepatic artery proper, and a small branch of the bile duct. And so this should tell you a little bit about how the liver operates. Remember, one of the roles of the liver is to manufacture bile. Well, where do you think the bile goes? It goes into these ductules, and it eventually is going to enter the gallbladder, which is on the underside of the liver, through the hepatic duct, right, which drains the bile straight into that little sac. And in the hepatic duct, what's going to happen to the bile is it's going to become concentrated. Branches from the arteries and the veins deliver blood to the sinusoids of adjacent lobules. As blood flows through the sinusoids, the hepatocytes absorb solutes from the plasma, and they secrete plasma proteins. And one of the many roles of the liver is that it manufactures albumin, which is a major osmotically active component in the plasma part of blood. Pressures in the hepatic portal system are low, averaging about 10 millimeters of mercury or less. This pressure change can increase markedly if blood flow through the liver is restricted by a blood clot or an organ damage, and this is called portal hypertension. This is one of the hallmarks of cirrhosis. What happens as scar tissue builds up is that you can occlude the blood flow through the organ itself, and then you end up with this portal hypertension as the scar tissue begins to accumulate. Okay, bile is pretty important stuff. Bile helps to emulsify fat in the small intestine. The liver secretes it into a network of narrow channels between adjacent hepatocytes. These canaliculi extend outward from the central vein. Eventually, they connect with the bile ductules, and this carries the bile into ducts in the nearest portal area. The right and left hepatic ducts collect bile from all the bile ducts of the liver lobes, and then these ducts unite to form the common hepatic duct, and that exits the liver and heads straight to the gallbladder. The bile in the common hepatic duct either flows into the common bile duct, which empties into the duodenal ampulla, which is an entrance right into the duodenum straight up, or it enters the cystic duct, where it enters the gallbladder and is now concentrated. The common bile duct is formed by the cystic duct and the common hepatic duct. The common bile duct passes towards the stomach, it turns, and then it penetrates the wall of the duodenum to meet the pancreatic duct at the duodenal ampulla. Okay, so we got to get these ducts sort of in our head. The duct immediately that branches off of the gallbladder is called the cystic duct. Joining the cystic duct is the hepatic duct. The cystic duct and the hepatic duct fuse and they form the bile duct, and it's the bile duct that is going to enter the duodenum. The liver is responsible for three categories of physiological function: metabolic regulation, hematological regulation, which is the control of blood chemistry, and bile production. In metabolic regulation, we look at the liver as the organ involved in regulating the composition of blood. All blood that leaves the absorptive surfaces of the digestive tract enters the hepatic portal system and flows into the liver to be cleaned. The hepatocytes extract nutrients and toxins from the blood before it reaches the systemic circulation through the hepatic veins. The liver removes and stores excess nutrients and corrects nutrient deficiency by mobilizing stored reserves or performing biosynthesis and then introducing those products into the blood plasma. The liver's regulatory activity affects carbohydrate metabolism. The liver has a huge role in stabilizing blood glucose at around 90 milligrams per deciliter. If blood glucose drops, the hepatocytes break down glycogen and release glucose into the blood to elevate those levels back within homeostatic limits. It's also involved in lipid metabolism. The liver regulates the circulating levels of triglycerides, fatty acids, and cholesterol. When these levels drop too much, the liver breaks down its reserves and releases them directly into the blood. So here we go. The liver as your body's kitchen, right? As your energy needs increase, the liver goes to the pantry and breaks down components of the pantry to make a tasty dish of fuel for the rest of the body. It's also involved in amino acid metabolism. It removes excess amino acids from the blood, and these molecules can be used to build proteins or converted into lipids or glucose for storage. And there's one other role for the liver that's sort of hidden here, which is the formation of a molecule called urea. Urea is formed by joining two molecules of ammonia to one molecule of carbon dioxide. This waste product then enters the blood and is eventually going to be removed by the kidneys, and we'll talk about that in chapter 26. Okay, it's also involved in waste product removal. When you convert amino acids to lipids or carbs, or when you break down amino acids to get energy, the liver has to strip off that nitrogen of the amino acid in a process called deamination. Now, where does that nitrogen atom go? It ends up being part of a urea molecule. Generally, we don't tolerate very high levels of straight ammonia, which is the other form of nitrogenous waste that enters the bloodstream, and that's why the liver has a major role in detoxification by forming this waste product, which, again, you don't want to accumulate to levels too high in the bloodstream, but urea is less toxic than ammonia. Okay, the liver is also involved in vitamin storage. The fat-soluble vitamins, remember those are A, D, E, and K, are absorbed from the blood and they're stored in the liver. They're called on when your diet has inadequate amounts of these vitamins. Now, a word about fat-soluble vitamins. Okay, fat-soluble vitamins are in a way good and in a way bad. I mean, they're good because they're primarily cofactors for enzymes that generate the energy we need to run the human body. Cofactors are basically like little tools that enzymes use to perform their job properly. So in that sense, we absolutely have to have a minimum daily allowance in our diet in order for these enzyme reactions to occur. They're bad in the sense that if you take too much of them, it's very easy for you to overdose, because remember that nonpolar molecules like to dissolve in the presence of other nonpolar molecules, and that can lead to toxicity. It's very easy, for instance, to overdose on any of these vitamins, and the result can end up being anything as severe as increased rates of cancer to toxic levels of these compounds in the adipose tissue that can end up actually killing certain tissues in the body. Okay, it's also involved in mineral storage. The liver converts iron reserves into ferritin and then stores this protein-iron complex for later use. Among those later uses include the production of hemoglobin. right? You need iron in hemoglobin in order for it to bind to the oxygen and actually carry it around the circulatory system. It also detoxifies drugs, and I think this is the great unsung role of the liver. It removes and breaks down circulating drugs and limits their effects. And there is a very important molecule that aids in this detoxification that exists in the liver. It's called cytochrome P450. And basically what that molecule does is it allows nonpolar toxins to dissolve easily in a water-based solution. The utility of that is that the kidneys can then excrete it from the body, and this is what we call metabolic turnover. So when you take a drug, even drugs that are quote unquote good for you, the liver has a job to do, which is to clear it from the body as soon as it possibly can. Now, in individuals that engage in recreational drug use or drink excessive amounts of alcohol, what you can do is you can overwork the liver. Let's take alcohol abuse as an example. The active component of alcohol is ethanol. Now, I can draw ethanol here for you right on this page. I got a little pen here. Ethanol looks like this. Okay, it's two carbon molecules bound together. All right, and you've got three hydrogens bound to the first carbon. Is it going to let me draw another hydrogen? I hope so. Whoop. Like that. So that's a CH3. I have a tough time drawing the rest of the H there. We'll try. Okay. And then over here, we have a hydroxyl group. Okay. And then you've got two more hydrogens bound. So this is the structure of ethanol. Okay. More hydrogen down there. Okay. Not the greatest drawing in the world, but you know, we'll work with it. Okay, whoops. Back up. Ah, shoot. Let me draw it up top. I have more room here. Let's make this white. This will be easier. Okay, here's ethanol again. Okay, so carbon, carbon, hydroxyl, and let's just put H3 here. You'll understand it. That's three hydrogens bound to a carbon, and then we've got a hydrogen up here and a hydrogen over here. All right, so that's ethanol. Now, what happens in the liver to ethanol is that ethanol is, among other things, a source of fuel. Ethanol can be used to generate energy. And what happens in the liver is an enzyme called ADH, alcohol dehydrogenase, will oxidize ethanol and turn it into a molecule called acetaldehyde. Okay, and acetaldehyde looks like this. It's a hydrogen, and that's another carbon, and then H3. Okay. All right. So what we've done here is we've oxidized the group that contains the OH. Right. We've turned that into a C double bond OH. That's an aldehyde group. The way you know that a carbon-containing molecule has been oxidized is that the number of carbon-hydrogen bonds in the molecule shrinks when you oxidize it. So count the number of carbon-hydrogen bonds. There's 3 here, there's 2 here for a total of 5, and over here you've got 3 plus 1 for a total of 4. That represents an oxidation. Now what happens to the molecule of acetaldehyde is that eventually it's converted into something called acetyl-CoA, which I won't draw out, it's just AC-CoA, and from there it enters a metabolic pathway that we'll talk about in the next chapter, which is called the Krebs cycle, which is a series of enzymatic reactions in the matrix of the mitochondria that are designed to, in a stepwise fashion, oxidize carbon-containing compounds such as pyruvate and acetyl-CoA and generate now high-energy molecules, NADH and FADH2, which are eventually going to be oxidized to generate large amounts of ATP. Okay, so this is going on in the liver. Now, the thing I want you to pay attention to is this molecule right here, acetaldehyde. Now, we've looked at pictures in the lab of tissues that have been preserved using a compound called formaldehyde. Now, what formaldehyde does is it pickles living tissue by producing large numbers of cross links between protein molecules and rendering the tissue dead, essentially, and intractable to decay. It's a way of preserving things. Pickling and salting were two of the early ways of preserving food if you were to travel across country without the aid of refrigeration. Okay, and that's why they were so popular. So what's happening in your liver is you're actually generating a compound here that is a first cousin to a very powerful pickling agent, formaldehyde. And you can probably guess what the effect is. The effect on living tissue, if this intermediate builds up too high, is the same as the effect of formaldehyde on living tissue, which is to basically kill it and pickle it, right? So you probably heard of people that drink a lot saying that they're pickled. Well, they're more pickled than they know. They're actually pickling their liver when they abuse alcohol. Now, that's bad all by itself because the liver performs a whole bunch of functions vital to life, one of which is drug inactivation, this roll down here that I was addressing earlier. And in individuals that engage in a lot of recreational drug use, drinking being among those drugs, the liver will eventually scar and begin to lose function over time. Even though it can regenerate in the face of damage, it can only sustain so much damage before it basically kicks the bucket. And so when these people get sick later in life, they find out that their doctors very often can't prescribe for them drugs that are designed to alleviate their disease symptoms because the liver wouldn't be able to detoxify that compound in the plasma. You'd basically destroy the liver and you'd kill the patient. Okay, so what starts out as seeming like a lot of fun ends up in the long run perhaps costing you your life. Okay, so thank your liver, give it a little pat while you're sitting there listening to this lecture, and say, I'm very happy that I have a functioning liver. Okay, what else does the liver do? Well, it's involved in blood regulation. The liver is the largest blood reservoir in your body. It gets about a quarter of the cardiac output. As blood passes through it, the liver phagocytizes pathogens and presents them to the lymphatic system. The Kupffer cells in the sinusoids are what do this. They engulf old and damaged red blood cells, cell debris, and pathogens, remove them from the blood. They are antigen-presenting cells, and as a result, they can stimulate the immune response. They're also engaged in the synthesis of plasma protein, the main one being albumin. Okay, well, albumin is important because it's an osmotically active, very large molecule, and it helps with our fluid balance. Very often, when the liver begins to fail, albumin isn't produced in as great a quantity, and that results in loss of blood volume and the leaching of fluid into peripheral tissues. And so you very often see edema in the belly. This is called ascites, and it accounts for the distended abdomens of people who are in stages of liver failure or are malnourished. Basically, they don't have enough protein in their diet to synthesize albumin. What else does it introduce into the blood? Clotting proteins and complement proteins. So I bet you can probably figure out that if you're in the early stages of liver failure, you may have problems with blood clotting and you may have problems with lipid and nutrient transport and hormone transport in the blood. Okay, all very important stuff. It also removes hormones. The liver is the primary site for absorption and recycling of epinephrine and norepinephrine, as well as insulin, thyroxine, and all the steroid hormones. It also absorbs cholecalciferol from the blood. The cells then convert it into an intermediate that is released directly into the blood. and that helps with bone mineralization. It also aids in removal of antibodies. It absorbs and breaks down antibodies after we don't need them anymore and releases their amino acids so they can be used either to build new proteins or maybe they can be used for energy. And again, it removes toxins. Lipid-soluble toxins in your diet are absorbed by the liver and stored in lipid deposits where they don't disrupt cell function as easily. Other toxins are removed from the blood and are either broken down or excreted in the bile or by the urinary system. It also synthesizes and secretes bile, which is excreted into the lumen of the duodenum. It's made up of water with minor amounts of ions and bilirubin, which is a pigment that comes from hemoglobin, as well as cholesterol and an assortment of lipids, and these collectively are known as the bile salts. So what does this bile do? Well, it helps us digest our fat. Most lipids in our diet are not water soluble. Why? They're nonpolar. They don't mix with water. They're greasy. Mechanical processing in the stomach makes big drops of fat and oil with a variety of lipids. Pancreatic lipase only acts at the surface of these lipid droplets. So what we have to do in order to effectively digest these triglycerides is we have to emulsify them, and that's what the bile accomplishes. It creates little tiny drops with a superficial coating of bile salts, and these drops increase the available surface area so that the lipase can break down the fat in your diet. Otherwise, you'd have a hard time processing the fat. And essentially what would happen is that it would work its way all the way through the small intestine and make it to the large intestine, and there it would produce GI upset because it would be osmotically active. It would also produce greasy stools and would lead in all likelihood to diarrhea. Okay. The gallbladder is a little pear-shaped organ on the underside of the liver that stores and concentrates this bile so that it's more effective. What happens as a result is we make sort of a super detergent that can be introduced into the duodenum at the command of the stomach. This sac is located in a fossa in the posterior surface of the liver's right lobe, right there. Okay. The gallbladder is split into three regions: the fundus, the body, and finally the neck. The duodenal ampulla opens into the duodenum at the duodenal papilla. This is a small mound of tissue that penetrates the duodenal wall. The muscular hepatopancreatic sphincter, also known as the sphincter of Oddi, circles the lumen of the common bile duct, and generally the pancreatic duct and duodenal ampulla as well. And what does this sphincter do? It opens and shuts at the command of the stomach and allows both the pancreatic juice and the bile salts to be introduced into the lumen of the duodenum. The major function of the gallbladder is to store bile, which is secreted continuously, about 1 liter a day, but it's released into the duodenum only under the stimulation of the intestinal hormone known as CCK, which is short for cholecystokinin. Without this hormone, the hepatopancreatic sphincter will remain closed, so the bile exiting the liver and the common hepatic duct won't flow through the common bile duct to the duodenum. Instead, it enters the cystic duct and is stored in the gallbladder. When chyme enters the duodenum, CCK is released. This relaxes the hepatopancreatic sphincter and causes the gallbladder to squeeze the bile into the small intestine. The amount of CCK secreted increases when the chyme contains a large amount of lipid. Well, why do you think that is? Well, because the more fat you've got, the more fat you need to emulsify. And so what happens is that we secrete this detergent in proportion to the fat content of your meals. Now, what do you think happens if CCK isn't properly generated, or if there's a breakdown between the production of CCK and the target organ here, which is the gallbladder? Well, the gallbladder will just sit there with bile inside it, and that bile will become more and more concentrated over time. And what can eventually happen is that you can form now little crystals inside the gallbladder known as gallstones, and they cause all kinds of havoc. So the gallbladder functions in bile modification. When it's full, it contains about 70 milliliters of bile. Bile is made up of primarily water bile pigments and salts, and it acts very similar to the way detergent acts on grease in your sink. The bile salts have a polar end and a non-polar end, and what they do is when they enclose the fat, they sort of build a bubble around it. The exterior of that bubble is made up of the polar component of the bile salts, while the long fatty acid tails of the bile salts interact with the grease, and that's what allows now the production of these emulsified surfaces. The composition of the bile changes while it sits in the gallbladder. Much of the water is reabsorbed, and the salts and other components of the bile get more and more concentrated. But if it gets too concentrated, crystals of insoluble material and salts form. These are gallstones. Small gallstones are not a problem as long as they can be flushed down the bile duct and excreted. But large bile stones can block the passage of bile into the duodenum. This produces extreme pain when you attempt to digest food as it comes from the stomach. And in further complications, sometimes what can happen with these gallstones is that they can wedge in the hepatopancreatic ampulla and block not only the bile salt entry into the duodenum, but also the entry of pancreatic juice into the duodenum. And that can lead to an inflammation of the liver, and it can also lead to an inflammation of the pancreas known as pancreatitis. So the liver is the center for metabolic regulation in the body, and it also makes the bile that's stored in the gallbladder and is ejected into the duodenum when the CCK signal is given. It's important for the efficient digestion of lipids. It breaks down large lipid droplets so that individual lipid molecules can be effectively attacked by lipase and broken into what? Fatty acids and glycerol. Those are the major components of triglycerides, which is the major fat in most of the food that you eat. A combination of mechanisms coordinates the activities of the digestive glands. These regulatory mechanisms are centered around the duodenum, where acids have to be neutralized and the appropriate enzymes need to be added. The neural mechanisms involving the central nervous system prepare the GI tract for activity. This again is parasympathetic innervation. Now remember what the nickname for the parasympathetic division of the autonomic nervous system is. It's the rest and digest division. They can also inhibit gastrointestinal activity through sympathetic innervation. We can also coordinate the movement of materials along the length of the digestive tract using the enterogastric, gastroenteric, and gastroileal reflexes. And basically, these are coordinated by the enteric nervous system. In addition, motor neurons that synapse in the digestive tract release a large variety of neurotransmitters. The intestinal tract secretes a variety of peptide hormones that also regulate its activity. They have multiple effects on different regions of the GI and in the accessory glandular organs. The duodenal enteroendocrine cells make secretin, which is released when the chyme arrives in the duodenum. Its effect is to increase the secretion of bile and buffers by the liver and the pancreas, and CCK, which is secreted when chyme hits the duodenum, especially if it contains a lot of lipids and protein. In the pancreas, CCK accelerates the production and secretion of all types of digestive enzymes. And why does that make sense? Well, because the food's coming, right? So what you want to do is prepare that region of the GI before the food arrives. Otherwise, you're not going to get the enzymes in the food to meet up effectively in order to promote chemical digestion, chemical breakdown of the molecules of your food. It also causes relaxation of the hepatopancreatic sphincter and contraction of the gallbladder, and this ejects bile and pancreatic juice right into the duodenum. GIP is secreted when fats and carbs enter the small intestine, and VIP is a hormone that stimulates the intestinal glands, dilating regional capillaries and inhibiting stomach acid production. Gastrin is made by the G cells in the stomach lining when they're exposed to incompletely digested protein. The function of gastrin is to promote the increased stomach motility and to stimulate the acid and enzyme production inside the stomach so that we can more effectively break down the food that's in there. Okay, so this is a hormonal signal that tells the stomach you're not quite done with the job yet. Enterocrinin is a hormone that's released when chyme enters the small intestine, and it stimulates duodenal mucin production. And what this does is this sort of protects the lining of the duodenum from the acidic secretions of the stomach, and it also lubricates the food as it goes through the GI. It takes about 5 hours for material to pass from the duodenum to the end of the ileum. So the first material to enter the duodenum after you eat breakfast may leave the small intestine about lunchtime. Along the way, the organ's absorption is enhanced by the fact that a lot of the mucosa moves around, those peristaltic waves that we were talking about last time we were together. The microvilli can be moved by their supporting microfilaments, the individual villi by smooth muscle contractions, and groups of villi by the muscularis mucosa, that thin layer of smooth muscle that lies just underneath the mucosa and the submucosa. Also, the plicae will move as the muscularis mucosa contracts and relaxes, as well as the muscularis externa, and those are the major smooth muscle layers which are located superficially to the muscularis mucosa, as well as the mucosa and the submucosa, but deep to the serosa. These movements mix the intestinal contents and they change the environment around each epithelial cell all the time. The point of all this is to make as much contact with the wall of the GI as you possibly can. Why? Because this enhances absorption into the bloodstream and into the lacteals. The less contact you've got with the lining of the GI, the less efficient the absorption is going to be. This is part of the effect that fiber has in your diet, is that it kind of increases GI motility and takes a little bit away from absorption. So this is why meals that are excessively high in fiber can actually impede nutrient uptake. It's also why a certain molecule compound that was made by P&G, this has been now, geez, probably been about 8 years already, called Olestra, was pilloried as a compound that would inhibit the absorption of nutrients into the GI, because essentially what Olestra was a molecule that couldn't be processed by any of the digestive enzymes in either the stomach or the small intestine or the oral cavity. And so what it did was it would coat the lining of the GI and would prevent nutrient absorption, specifically of fat-soluble components, but it really inhibited nutrient absorption across the board, because it basically made a layer of artificial grease that would block the movement of nutrients into the bloodstream. Now, if you're trying to lose weight, that's an effective way to do it. Okay, if you block nutrient absorption, guess what? Fewer calories are going to get into the blood, fewer calories are going to get to the liver, and less lipid is going to be stored as adipose tissue in the long run. The bad side of this is that it promotes explosive diarrhea, which was an unfortunate side effect of this particular compound in certain individuals, and it also inhibited the absorption of vitamins across the GI. So it was taken off the market. The initial product that was promoted as made with Olestra were the WOW chips, and they had adverse effects on certain individuals, and so that product line was pulled. But I bet you didn't know that Olestra is still out there on the shelves as part of the Pringles fat-free chip brand called Light, and people are still buying that product and eating it. So how much of it was media hype and how much of it was actual effects, it's hard to say. Okay, now we get to the end of the trip, the large intestine. We'll talk about the histological structure, and we'll talk about the different regions of the large intestine and explain the significance of the large intestine in nutrient absorption. Large intestine looks a lot different than the small intestine. For one thing, it ain't nearly as long and it doesn't have nearly as much surface area. And the reason is it doesn't need it. By the time the food gets to the large intestine, the nutrient value has been extracted mostly, and what you're left with are products that we don't have the enzymatic activity to process and absorb so that we can extract usable energy out of it. That is left to the inhabitants of the large intestine called the bacteria. They have the enzymatic machinery to break down our indigestible portion of food, primarily fiber, and in turn they produce components that are useful to our physiology, such as vitamin K, which is important in blood clotting. It's a horseshoe-shaped organ that begins at the end of the ileum and ends at the anus. It's inferior to the stomach and the liver, and it frames the small intestine. Its primary job is to reabsorb water and to compact the intestinal contents into fecal material. It's also important in the absorption of vitamins that are liberated by bacterial activity, such as vitamin K, and it's involved in the storage of fecal material prior to defecation. It has an average length of about 5 feet. It's about 3 inches wide. We can split it into 3 parts: the cecum, which is the first portion, the colon, the next portion, and the largest and longest portion, and finally the rectum, which is the last 6 inches and marks the end of the GI tract as it enters the anus. The material that comes from the ileum enters the cecum through the ileocecal valve. By the time it gets there, hopefully all the nutrient content of the food has been absorbed into the bloodstream. The cecum stores material from the ileum and begins the process of compaction. Notice that at the inferior end of the cecum is this little sucker down here. That's your appendix. The appendix is a dead-end sac filled with lymphatic tissue, and occasionally, in some people, it can become impacted with fecal material, and that can result in the production of gas as the bacteria inside the sac metabolize, which can cause the sac to distend, pressure to build up, and then, in an unfortunate turn of events, it can burst. A ruptured appendix is a medical emergency because the bacteria then are inside the peritoneal cavity where they can produce peritonitis and possibly septicemia. Okay, so individuals reporting of severe pain at McBurney's point, which is the midway point between the ileum and the navel, should go to the emergency room toot suite because this could be appendicitis. Okay, the cecum stores materials from the ileum and begins the process of compaction. The appendix is attached to the posterior medial surface. It's about 4 inches long and it can be of several different shapes. The mesoappendix connects the appendix to the ileum and the cecum. Okay, so that would be this tissue right here. The mucosa and the submucosa of the appendix are dominated by lymph nodules, and the primary function of the appendix is as an organ of the lymphatic system. But you can live perfectly fine without your appendix. You don't need it to live. This is why an appendectomy isn't something that you should fret too much about. The colon has a larger diameter and a thinner wall than the small intestine. The distinctive features include the wall of the colon, which forms a series of these little, they almost look like, I don't know, bubbles along the length of this tube. Those are called haustra. Okay? The haustra are formed by the contraction of this little strap of smooth muscle called the teniae coli, which is the muscularis externa of the large intestine. The thing is that it has atrophied to the point where it is this extremely weak, insubstantial piece of tissue. Okay, so it doesn't have a lot of power in there, but it doesn't have a very long trip, so hopefully it can do the job. Generally it can. The haustra permit expansion and elongation of the colon. The separate longitudinal bands of smooth muscle, the teniae coli, run along the outer surface of the colon just deep to the serosa. They correspond to the outer layer of the muscularis externa in other portions of the GI. Muscle tone in the teniae coli create the haustra. So if you got good teniae coli, you can really move that fecal material along. If they're weak, then you're going to have some trouble. The serosa of the colon contains a lot of sacs of fat called epiplotic appendages. Epiplotic appendages. You can see these little yellow sacs all along the Tenier coli if you look very carefully. We can split the colon into 4 regions: the ascending, the transverse, the descending, and the sigmoid colon. The ascending colon begins at the superior border of the cecum and it ascends along the right lateral posterior wall of the peritoneal cavity to the inferior portion of the liver. Okay, so the liver be right about there. Okay, there the colon bends sharply to the left at the right colic flexure, and this marks the end of the ascending colon and the beginning of the transverse colon. The transverse colon curves anteriorly from the right colic flexure and crosses the abdomen from right to left. It's supported by the transverse mesocolon and is split from the anterior abdominal wall by the greater omentum, that apron of fat that hangs in front of both the large and small intestine and provides padding, insulation, protection, and is a source of fuel, also known as your beer gut. Okay. As the transverse colon reaches the left side of the body, it passes under the greater curvature of the stomach. Near the spleen, the colon makes a 90-degree turn at the left colic flexure, and now it becomes the descending colon. Okay, so right colic flexure, left colic flexure, and now descending colon. The descending colon proceeds inferior along the person's left side until reaching the iliac fossa, formed by the inner surface of the left ilium. The descending colon is retroperitoneal, meaning it's behind the peritoneal membrane and firmly attached to the abdominal wall. At the iliac fossa, the descending colon curves and the sigmoid flexure becomes the sigmoid colon. This sucker right here. Sigmoid means S-shaped. The sigmoid flexure is the start of the sigmoid colon. Okay, there's the sigmoid flexure, there's the sigmoid colon. It's an S-shaped segment that is only about 6 inches long. It lies posterior to the urinary bladder, suspended from the sigmoid mesocolon. It empties directly into the rectum. The large intestine gets blood from the superior and inferior mesenteric arteries. Remember those branches off the descending aorta, which is termed the abdominal aorta in this region. Venous blood is collected from the large intestine by the superior and inferior mesenteric veins. Now we get literally to the end of the trip, the rectum. It forms the last 15 centimeters of the digestive tract and is an expandable organ for the temporary storage of feces. The movement of fecal material into the rectum triggers the urge to defecate. The last portion of the rectum, called the anal canal, contains small longitudinal anal columns. Okay, so you can see here the anal columns. All right. The anus or anal orifice is the exit of the anal canal. There the epidermis becomes keratinized and identical to the surface of the skin. Why do you think that is? Well, think about it. There's mechanical force here, okay? And what you'd want is to have a substantial resistance to this mechanical force, or else it could bleed. And if you bleed in this area, you're in trouble because there's a lot of bacteria down there. The circular muscle layer of the muscularis externa in this region forms the internal anal sphincter, which is smooth muscle, and this is not under voluntary control. The external sphincter is skeletal muscle. It guards the anus, and its muscle fibers encircle the distal portion of the anal canal. Okay, so there's the external anal sphincter, and there is the internal anal sphincter. This sphincter consists of skeletal muscle, and we control it. Although the diameter of the colon is about 3 times that of the small intestine, its wall is much thinner. The major characteristic of the colon is the lack of villi, right? We don't need a lot of surface area here because we're not doing a lot of nutrient absorption. There's also a lot of goblet cells which produce mucus that help to lubricate the fecal material so that it can pass. Also, we have a lot of distinctive intestinal glands all along the surface. This is a cartoon of the lining of the large GI. and this is an actual histological section taken through here. So you can see the mucosa, right? The submucosa down here, the muscularis externa, which is almost non-existent here, and then finally Tenier coli, which is going to be, you can see it here in this diagram, all right? You can't really see it so much in this section. The glands in the large intestine are deeper than in the small intestine, and they're primarily goblet cells. The mucosa does not produce enzymes. Any digestion that occurs is the result of enzymes introduced into the small intestine or from bacterial activity. The mucus provides lubrication as the fecal material becomes dry and compact, and large lymphoid nodules are scattered all throughout the lamina propria and the submucosa. And I can't think of a better place for them because you're talking about a lot of bacteria in this region. We don't want that to get into the blood. The muscularis externa of the large intestine is unusual because the longitudinal layer has been reduced to the muscular bands known as the Tenier coli, right there. Okay, so let's talk a little bit about the physiology of the large intestine. About 10% of the nutrient absorption underway in the digestive tract takes place here. It also prepares fecal material for ejection from the body. Reabsorption of water is an important function of the large intestine. Although about 1.5 liters of material enters the colon each day, only about 200 milliliters of feces are ejected. So that's a substantial reduction in volume. The large intestine also absorbs a number of other substances that remain in the feces or were secreted into the GI along its length. Most of the bile salts that enter the large intestine are reabsorbed in the cecum and transported in blood to the liver for their reconstitution as bile. Okay, so there's some recycling going on here as well. Vitamins are organic molecules that are important as cofactors or coenzymes in a lot of metabolic pathways. The normal bacterial residents of the colon generate 3 vitamins that supplement our dietary supply. Vitamin K, we've already discussed. It's a fat-soluble vitamin that the liver needs for synthesizing the clotting factors, including prothrombin. Intestinal bacteria produce about half your daily vitamin K needs. It also helps in the absorption of biotin, which is a water-soluble vitamin important in various reactions, notably in glucose metabolism. So thank you, bacteria, for these 2 additions to our vitamin array. And pantothenic acid, which is a water-soluble vitamin required in the manufacturing of steroid hormones and neurotransmitters. In the large intestine, bacteria convert bilirubin to urobilinogen and stercobilinogens, which have a brown color, hence the brown color of feces. Some urobilinogens are absorbed into the blood and excreted in the urine. The urobilinogens and stercobilinogens remaining in the colon are converted to urobilins and stercobilins when they hit oxygen. The bacterial action breaks down peptides that remain in the feces, and it generates ammonia in the form of soluble ammonium ions, and indole and skatole, which are nitrogen-containing compounds that are primarily responsible for the odor of fecal material, and hydrogen sulfide, which is a gas that produces a rotten egg odor. Basically, any molecule that contains sulfur is going to have a very strong, disagreeable smell. Indigestible carbs are not altered by intestinal enzymes, so they arrive intact at the colon. These molecules provide a reliable nutrient source for the bacteria, and they're able to break this down, and in the act of doing so, produce gas. Okay, so this is where our intestinal gas comes from, the bacterial activity. So anything that supplements bacterial growth is going to increase the amount of intestinal gas. So think about compounds that you eat that have a lot of carbs in them, such as beans. Okay, the gastroileal and gastroenteric reflexes move material into the cecum while you eat. Movement from the cecum to the transverse colon is slow, and it allows hours for water to absorb back into the body, and this turns the liquid material into a paste. Peristaltic waves move the material along the length of the colon, and segmentation movements, called haustral churning, mix the contents of the adjacent haustra. Movement from the transverse colon through the rest of the large intestine results from powerful peristaltic contractions known as mass movements, and these occur or should occur a few times each day. The stimulation of distension in the stomach and the duodenum sends commands to the large intestine and help propel the fecal material along. Distension of the rectal wall triggers the defecation reflex, which involves two positive feedback loops. Both are triggered by the stimulation of stretch receptors in the walls of the rectum. The first loop is a short reflex that triggers a series of peristaltic waves in the rectum that move the feces to the anus, and the second loop is a long reflex coordinated by the sacral parasympathetic system. It stimulates mass movements that push feces towards the rectum from the descending colon and the sigmoid colon. An interruption in these signals can result in a stall in the movement of the fecal material through the colon, which can result in constipation. Rectal stretch receptors also trigger two reflexes important to the voluntary control of defecation. One is a long reflex that's mediated by parasympathetic innervation within the pelvic nerves. This reflex causes the relaxation of the interanal anal sphincter, the smooth muscle sphincter, which controls the movement of feces into the anal canal. The second is a somatic reflex that stimulates the immediate contraction of the external anal sphincter, which is a skeletal muscle. The motor commands are carried by the pudendal nerves. The elimination of feces requires that both the internal and external sphincters be relaxed at the same time. Otherwise, the fecal material is not going to pass. But the two reflexes just mentioned open the internal sphincter and close the external. The actual release of feces requires a conscious effort to open the external sphincter. In addition to opening the external sphincter, consciously directed activities such as tensing the abdominal muscles or making expiratory movements while closing the glottis elevate the intra-abdominal pressure and help force the material out of the rectum. This is called the Valsalva maneuver, and it's used in the expulsion of feces. It's also used during pregnancy. Okay? We'll specify the nutrients that are required by the body, and we'll describe the chemical events responsible for digestion of organic nutrients. And we'll also describe the mechanisms involved in the absorption of organic and inorganic nutrients. So what we're going to get into here now is some chemistry. A typical meal contains carbs, proteins, lipids, water, electrolytes, and vitamins. The GI handles each component in a different chemical fashion. Large organic molecules have to be broken down by digestion before we can absorb them. Water, electrolytes, and vitamins can be absorbed without processing, but special transport mechanisms may be involved. Food contains large organic molecules. Many of them are insoluble in water. The GI first breaks down the physical structure of the ingested material and then proceeds to disassemble the common component molecules into tinier fragments. The molecules released into the blood are absorbed by cells and either broken down to provide energy for the synthesis of ATP or are used to synthesize carbs, proteins, and lipids. GI enzymes break the bonds between component molecules of carbs, proteins, lipids, and nucleic acids in a process known as hydrolysis, which uses a water molecule to split a polymer into its monomeric components. The classes of digestive enzymes differ with respect to their target. Carbs break the bonds between simple sugars, proteases split the linkages between amino acids, and lipases separate the fatty acids from the glycerides. Again, a disassembly line. Digestive enzymes in the salivary glands, tongue, stomach, and pancreas are mixed into the ingested material as it passes along the tract. These enzymes break down large carbs, proteins, lipids, and nucleic acids into smaller fragments, which in turn typically have to be broken down further before they can absorb into the blood. The final steps involve brush border enzymes. These line the small intestine, and they're embedded in the microvilli. Nucleic acids are broken down into their nucleotides, and brush border enzymes digest these into sugars, phosphates, and bases that are absorbed by active transport. What happens to the carbs? Well, complex carbohydrates, such as polysaccharides and starches, are processed in two steps. One step involves carbohydrases made by the salivary glands, which begin the process. They're not horribly effective, but they get the job going. And the pancreas, which is a more extensive dose of carbohydrate breakdown. Pancreatic amylase is one of these enzymes. The other are the brush border enzymes that break down, for instance, disaccharides into their monomeric components. Digestion of complex carbs involves two enzymes, salivary and pancreatic amylase, that function effectively across a fairly wide pH. Carb digestion begins in the mouth during chewing, although the action of amylase from the parotid and the submandibular salivary glands isn't as extensive as the pancreatic activity. Again, it begins the job. Salivary amylase breaks down starches, producing a mix composed primarily of disaccharides and trisaccharides. Amylase continues to digest the starches and glycogen in the food for about 2 hours before the stomach acids render the enzyme inactive. Once we're in the small intestine, we can talk about what the brush border enzymes do. Prior to absorption, disaccharides and trisaccharides are fragmented into monosaccharides by brush border enzymes of the intestinal microvilli. The enzyme maltase splits bonds between the two glucose molecules of maltose, producing 2 molecules of glucose. And sucrase breaks the disaccharide sucrose into glucose and fructose, which is another monomeric sugar. Lactase hydrolyzes the disaccharide lactose into a molecule of glucose and one of galactose. It is a hereditary lack of this enzyme that is responsible for the condition known as lactose intolerance. Now, if we think about this for a moment, you can imagine what the consequences would be. If this disaccharide isn't broken down into its monomeric components, it will not absorb into the blood. If it does not absorb into the blood, it will remain in the GI until it reaches the large intestine, where bacteria are waiting for this extremely rich energy source. The result is loose stool because, again, the lactose is osmotically active, and also the production of gas, distension, and pain. So how do we avoid this if we are lactose intolerant? Well, we either supplement the GI with an external source of lactase in caplet form. These are called probiotics. Or we avoid the ingestion of lactose, which is primarily found in dairy products. How about the monosaccharides? What happens once we get them? The intestinal epithelia absorbs the monosaccharides by facilitated diffusion and co-transport. Both methods involve carrier proteins. Facilitated diffusion and co-transport differ in 3 major ways. The intestinal epithelium, which absorbs these monosaccharides, use facilitated diffusion. Facilitated diffusion moves only one molecule or ion through the cell membrane. Co-transport moves more than one molecule or ion through the membrane at the same time. In co-transport, the transported materials move in the same direction down the concentration gradient for at least one of the transported substances. This does not require an energy source. Okay, this is something that will happen spontaneously, driven by the difference in concentration on either side of the membrane. Facilitated diffusion does not require ATP, although co-transport by itself does not consume ATP. The cell often expends ATP in order to preserve homeostasis. Facilitated diffusion will not occur if there is no opposing concentration gradient for the molecule that it's trying to move. By contrast, co-transport can occur despite an opposing concentration gradient for one of the transported substances. Basically, what it can do is it can hitch a ride on an existing gradient for another molecule in order to drive a second molecule against its concentration gradient either into or out of the cell. Okay, and this is a phenomenon called secondary active transport because it really, at its core, gets its energy from an active transport gradient that was established by a different set of membrane-bound enzymes. The cotransport system responsible for the uptake of glucose also brings sodium into the cell. This passive process resembles facilitated diffusion, except that both the sodium ion and a glucose molecule have to bind to the carrier protein before they can move into the cell. Comparable cotransport mechanisms exist for other sugars and for some amino acids. Although these mechanisms deliver valuable nutrients to the cytoplasm, they also bring in sodium ions that have to be ejected by the sodium-potassium ATPase. This is a pump. It's found in every cell in the body. It consumes anywhere between 2/3 and 3/4 of our energy, but it's important in maintaining membrane potential and obviously for these transport mechanisms. Simple sugars that are transported into the cell at its apical surface diffuse through the cytoplasm, and they reach the interstitial fluid by facilitated diffusion across the basolateral surfaces. These monosaccharides diffuse into the capillaries of the villus for eventual transport to the liver in the hepatic portal vein. And what happens there? It's either stored as glycogen or fat, or it's passed on to the rest of the body. Lipid digestion is a little different. It involves lingual lipase from the glands of the tongue and pancreatic lipase from the pancreas. The most important and abundant dietary lipids are triglycerides, which consist of 3 fatty acids attached to a single molecule of glycerol. The lingual and pancreatic lipases break off 2 of the fatty acids, leaving a monoglyceride. Lipases are water-soluble enzymes, and lipids tend to form droplets that exclude water molecules because of their hydrophobicity. As a result, lipases can attack only the exposed surface of the lipid droplet. Lingual lipase begins breaking down triglycerides in the mouth and continues until we reach the stomach, but the lipid drops are so large and the available time so short that only about 20% of the lipids have been digested by the time the chyme gets to the duodenum. So what are we going to do about this? Continue the digestion. And this is where we call the bile salts in to help us out. Bile salts improve chemical digestion by emulsifying the lipid drop, so it increases the surface area available for the lipases to act. And what this does is it further breaks down the triglycerides into monoglycerides and fatty acids. The intestinal cells then make new triglycerides from the monoglycerides and fatty acids, and these, in company with absorbed steroids, phospholipids, and fat-soluble vitamins, are coated with proteins, creating complexes known as chylomicrons. The intestinal cells then secrete the chylomicrons into the intestinal fluid in a process known as exocytosis. And so this is what we call our bad cholesterol. Okay? These are low-density lipoproteins, and what they indicate is that you are in a fat storage mode. If you have a lot of low-density lipoproteins and chylomicrons, you've got a lot of fat in the blood, a lot of fat in the lacteals. And this tends to produce a condition called arteriosclerosis, where the lumen of the blood vessels will narrow as plaque deposits on the endothelial lining. And what this does is it increases your blood pressure, and it also increases the risk of a vascular accident as a result of bursting a capillary bed, but you can also throw these plaques, producing emboli, which can lodge in the brain and cause a stroke, or lodge in the heart and cause a heart attack. Okay, what about the water? Cells can't actively absorb or secrete water. Water moves by osmosis. All of the movement of water across the lining of the GI, as well as the production of glandular secretion, involves passive water flow down osmotic gradients. How about the ions? Osmosis doesn't distinguish among solutes. All that matters is the concentration gradient. To maintain homeostasis, the concentrations of specific ions need to be closely regulated. The rate of sodium ion absorption by the digestive tract is increased by the hormone aldosterone, our salt-retaining hormone, which is made by what organ? I'll give you 10 seconds. And I hope you came up with the adrenal cortex, specifically the outer layer of the adrenal cortex. The rate of sodium ion absorption by the digestive tract under the influence of aldosterone is closely regulated, right? This is a major component in maintaining what we call membrane potential. Calcium ion absorption involves active transport at the epithelial surface, and the rate is accelerated by the hormone known as parathyroid hormone. Okay, now what organ do you think secretes parathyroid hormone? I hope you came up with the parathyroid gland. Okay, now one more question. Where is the parathyroid gland located? And I hope you came up with the posterior side of the thyroid gland, which lies just underneath the thyroid cartilage of the larynx. Okay. Calcitriol also affects the rate of calcium transport. As other solutes move out of the lumen, the concentration of potassium ions increases. These ions can diffuse into the epithelial cells along their concentration gradient. The absorption of magnesium and iron and other cations involves specific carrier proteins. The cell has to use these in order for ATP to be active, and it also has to use ATP to transport these across the cell membrane. The anions chloride and iodide, as well as bicarbonate and nitrate, are absorbed by diffusion or carrier-mediated transport. Phosphate and sulfate ions enter epithelial cells only by active transport. How about the vitamins? These are organic compounds required in tiny amounts, but they're very important. As we said, they're coenzymes and cofactors. They help enzymatic reactions go. They're like little tools. There's two major groups of vitamins, the fat and the water solubles. The fat solubles are the A, D, E, and K. The water solubles are the B complex vitamins and vitamin C. The 9 water soluble vitamins, the B vitamins, are found in milk and in meats. Vitamin C is found primarily in citrus fruits. All but one of the water soluble vitamins are easily absorbed by diffusion across the digestive epithelium. Vitamins can't be absorbed by the intestinal mucosa in normal amounts unless the vitamin has been bound to intrinsic factor, which is a glycoprotein secreted by the parietal cells of the stomach. And the vitamin we're talking about here is vitamin B12. Okay? A lack of intrinsic factor results in a lack of vitamin B12 in the blood, and vitamin B12 is vital for the synthesis of hemoglobin, and this results in a condition known as pernicious anemia. Okay? Pernicious anemia. Okay, as you probably guessed, aging does not improve the ability of the digestive system to operate. We'll summarize the effects of aging on the GI. Essentially, normal digestion and absorption occur in elderly individuals, but many changes in the GI parallel age-related changes we've already talked about in other systems. The rate of epithelial cell division slows down as we age. The digestive epithelium becomes more susceptible to damage by abrasion, acids, and enzymes. Smooth muscle tone drops. The general motility decreases, and peristaltic contractions are weaker as a result of a decrease in smooth muscle tone. What else happens? Cumulative damage becomes more apparent. A familiar example is the gradual loss of teeth due to dental caries. These are cavities, or to gingivitis. It can also involve internal organs. Toxins such as alcohol and toxic chemicals absorbed by the GI are transported to the liver. Cancer rates increase. They're most common in organs in which stem cells divide to maintain epithelial cell populations. The reason for this is that every cell division represents a possibility for a change in the DNA which could produce a cancer cell. Rates of rates of colon cancer and stomach cancer rise with age. Oral and pharyngeal cancers are particularly common among elderly smokers. There are also changes in other systems that have indirect effects on the GI. The decline in olfactory and gustatory sensitivity can lead to dietary changes, and this can affect the entire body. Okay, this brings us to the end of Chapter 24. This material will be covered on the upcoming exam, so please watch this podcast and take notes as you go along. If you have any questions, please get in touch with me, and I will see you in class.